is Dr. Hall at UW Stout. I just want to show you a few images of what our slides of the kidney look like in the lab, so renal histology. First, we're going to start off looking at the renal medulla, specifically at the pyramids, because in the renal pyramids, where we're going to find the loops of Henle as well as the collecting ducts of our nephron. So when we look at low power, we see lots of cells, right? It looks really pink, but you see these open spaces. And so these open spaces are the insides or the lumens of the collecting ducts. We have a nice big one here, right? So this is a collecting duct. We've kind of sliced through it longitudinally, and we can see that lumen. So let's look at higher power. We come in closer. Again, here is the collecting duct. And you can see the lumen. And look at how the wall of the collecting duct is made of simple cuboidal epithelium. These little square cells, they look to me like little marshmallows, right, lining the sides of the collecting duct. Here's another image. Again, you can see the lumen of the collecting ducts, and you can also see that simple cuboidal epithelium. Now, in some cases, these look a little bit more rectangular than square, and that just has to do with the angle at which they got cut, right? So if you cut through a cube diagonally, it'll look a little bit like a rectangle, but you can see here that nice square shape of these simple cuboidal epithelial cells. Here's another image. I just think this is so pretty. It's just lovely, all those simple cuboidal cells. All right, let's take a look at the renal cortex now, which is that outer rind of the kidney, where we're going to find the glomeruli, the arterioles, and both the proximal and distal convoluted tubules are all located out peripherally in the renal cortex. So it looks very different, even on very low power. So what we're seeing here, as has been labeled, this is the glomerulus, that ball of capillaries. And then all these other things, these round things that we see, these are tubules, proximal and distal convoluted tubules that happen to get cut on end. So we can see that lumen or that opening on the inside of them. Here's another glomerulus down here. Another one over here, right? So all of these glomeruli with their proximal and distal convoluted tubules all packed in very closely. Remember, the kidney has over 1 million nephrons. So let's zoom in to higher and look at this. It's amazing what we can see. So this is that little nest of the glomerular capillary. Oh, look, we can even see some individual erythrocytes. You see that? Those little erythrocytes there inside that capillary, super cool. So this is ball of capillaries, that glomerulus, and then we have Bowman's capsule around the outside, and we have that capsular space. So our filtrate is going to be squeezed out into that capsular space, and then it's going to drain into, of course, the proximal convoluted tubule. We didn't get this section so that we can see that proximal convoluted tubule kind of coming from the capsular space in this particular view. But boy, we can see a lot of other things, so that's pretty neat. This is another view of, again, we have that Bowman's capsule, that capsular space here, the glomerulus, that ball of capillaries, and in this view, in this particular one, we can also see these really interesting cells over here. This is that juxtaglomerular apparatus. Remember, juxta means next to, so juxtaglomerular apparatus. It's not a very inspired name, but we just call it the JGA for short. And so these are those cells that are closely adjacent to the afferent arteriole. They're part of a distal tubule, but they're very close to the afferent arteriole, and they are going to be sensing the blood pressure in that afferent arteriole as well as the sodium content. And if either of those two things is low, the JGA cells are then going to secrete the enzyme renin, which will cause a series of events to unfold, resulting in the production of aldosterone, which is a hormone that then tells the kidney to reabsorb more sodium, which causes water to passively follow due to the osmotic activity of the sodium. And so you end up reabsorbing more sodium and more water, which increases your blood volume and therefore your blood pressure. I think I have another slide. Yep. So here's another one. Again, you can see that glomerulus and you can see the juxtaglomerular apparatus cells right here. You might say, but Dr. Hall, I don't see the afferent arteriole. I know, it's not in this particular view, but I did find one. Well, not this one. That's the JGA again. Aha. So here we go. And look at all these little erythrocytes, right? Little tiny, they're just packed in here, little erythrocytes inside an arteriole. So I wasn't able to find a section that had both arteriole and the JGA together in it, but they do sit adjacent to each other so that those JGA cells can sense sodium and blood pressure. All right, so that's it for renal histology. Please contact me with any questions that you have.